and welcome to this video on the applications of gene cloning. There are many applications of gene cloning. However, in this video I will be discussing few. Gene cloning is a process in which we insert a section of DNA into a cell and then let the cell undergo mitosis, creating copies of the genetic information. This illustration shows the process of gene cloning. Gene cloning had an immense contribution in medicine and science. Genetic cloning allows us to study genetic disorders in a lab environment without having to endanger the patients themselves. In medicine, at the simplest level, molecular biologists routinely make clones of DNA, the molecular basis of genes. DNA fragments containing genes are copied and amplified in a host cell, usually a bacterium. This process had led to the production of important medicines such as insulin to treat diabetes, tissue plasminogen activator to dissolve clots after a heart attack. In forensics, if a crime has been committed and a small amount of DNA has been found on the scene, scientists in the forensics department need to study it. These tiny amounts cannot be used directly by the scientists, so they need to make more copies of DNA so they have enough to work with. This is called gene amplification. Gene cloning has contributed hugely to agricultural research and its application. It has mainly been applied in agriculture to develop resistance to pests and diseases and improve nutritional quality of crops. Cloning could result in the mass production of genetically modified cattle that secrete valuable drug into their milk. For example, milk that has human clotting factor IX can treat people with hemophilia, milk with insulin to treat people with diabetes, interferon to treat people with viral infections. In conclusion, cloning can make creation of transgenic animals to be mass produced. Large numbers of transgenic animals could produce vast quantities of needed drugs and other useful substances more efficiently and at a much lower cost.